And so the last thing we're going to talk about in Chapter 1 is the second golden age of microbiology. And so to start talking about the second golden age, I want to first talk briefly about what molecular biology is. So molecular biology is looking at the genes that make up individuals. So what makes up a cell? What molecules? And what is it that is passed on from parent to offspring that is sent from one organism to another? So before we even really knew anything about what cells, what was inside of cells and what was being sent from parent to offspring, Gregor Mendel, came up with rules, or I'll say rules that help us understand how we inherit certain genetic characteristic from parents to offspring. So he's known as the father of genetics. He is the monk who played with pea plants, and he was able to figure out all kinds of really cool things just by looking at peas and crossing different peas. So let's move up to the 1900s now. And in the about 1918 to around 1920, Frederick Griffith came up with this idea of transformation. And he said that transformation occurs when a dead cell can transfer information to the living cell, and the living cell can then use that information. So what it is, or what we now know, is that DNA from a dead cell is taken in by a living cell as naked DNA in a solution. And so here is his process, or his experiment. His hypothesis: material in dead bacterial cells can transform living cells. And so right here you have these cells that are called S cells. They were a type of streptococcus, and they were called S cells because they were smooth. They had a capsule around them. And when he infected mice with these S bacterial cells, the mice got sick and died. Here we have the same species, but these are called the rough, or the R strain of the streptococcus. And when he infected the R strain, the bacteria did not kill the cells, or the mice. So then he thought, okay, well, what's going on? That's so strange. The smooth kill, so the capsule must be causing the death because the cells themselves didn't do anything. So then he tried to just put the capsule in. So dead cells, but they are the smooth strain. And he infected the bacteria, or he infected the mice with the dead, smooth bacteria. And the mice lived. He was like, what is going on? This is so bizarre. So he thought, okay, I know that the living cells don't kill and the smooth dead cells don't kill, but the smooth living cells do. So what if I give them smooth cells with dead bacteria and rough cells that are living? So I gave them the living bacteria and the smooth capsules. And what happened was the mice died. So after each of these experiments, they then did an autopsy on the mice. So they either took the dead mice or they killed the mice, and they looked for the bacteria. So in the first bacterial species here, with the S strain, you found living S bacteria. In the second one, the mouse lived. So when they did the when they looked at the tissues, they found no S bacteria. They did find a few of the living cells, but that was it. The living R, but no living S. So when they looked here with the dead S strain, there was nothing inside the mouse. But when they took living rough and dead smooth, they found both living rough and living smooth bacteria. And so the rough bacteria here took information from the dead encapsulated cells and were transformed into the smooth cells. That's the idea. So that was in like the 1920s. And now we move to the 1940s, and Avery, McLeod, and McCarty did an experiment that highly suggested that DNA was the genetic material. So what their experiment was, they looked at those S cells, heat-killed smooth cells. And they then put the smooth cells into a solution. They added proteases to one of the experimental tubes. And proteases are going to break down any proteins. They added RNases into another solution. And what an RNase will do is they are going to break down any RNA. And then they added DNase to a third tube, culture tube. And the DNase is going to break down any DNA. Once that was done, they then added rough cells, living rough cells, to each of the solutions. And in the solution that had proteases added, you had smooth cells appearing, that living smooth cells that they were able to isolate. In the no RNA one, you had, again, living smooth cells isolated. But in the one where there was no DNA, there was only rough cells able to be isolated. There was no living cells able to be isolated. So that supported the idea that the genetic material of the cell was DNA. So Hershey and Chase then built on that and basically proved that, yes, DNA is the genetic material. And so in their experiment, they used radioactive bacteriophages. Remember when I said viruses can infect everything, including bacteria? A bacteriophage is a bacterial virus. And so they used radioactive bacteriophages. They used two types of radioactive bacteriophages. They used one bacteriophage that had radioactive sulfur, and though sulfur is found in proteins, so that would be in the protein capsid. They used another bacteriophage, and in that one they used radioactive phosphorus. So phosphorus is found in the genetic material of the virus. And so here you have the protein capsid is radioactive, and down here you have the genetic material. The DNA inside is radioactive. So they infected bacteria in a solution. They added the bacteriophage with bacteria in a solution, allowing the viruses to infect the bacteria. They then used a blender, super, fancy laboratory material, that mixed everything up and spun everything out a little bit. Then they that allowed it to be centrifuged, or that blended it. I'm sorry, that blended it so that the bacteria and the viruses were moved apart. They centrifuged everything, and bacteria are heavier than the viruses, And so the bacterial cells will actually fall into what we call a pellet. And the top will be the broth with any viral capsids, okay? And so at the end of the first experiment with the radioactive capsid, we found the radioactivity was in the solution and not in the bacterial pellet. And the second one, when they had the radioactive DNA and they allowed the viruses to infect the bacteria, they then got rid of the viral capsids from the bacteria using the blender. They centrifuged it, producing a solution with a pellet at the bottom. And in this case, the pellet was what was radioactive. The capsids were not. So the information that was radioactive, the DNA actually went into the bacteria, but up here, the radioactive capsid did not go into the bacteria. So that allowed us to undeniably determine that DNA and not the protein was the genetic material that transforms organisms. So now we know that DNA is the genetic information. We know how diseases are caused. We know that bacteria can cause diseases, and we know which bacteria cause which diseases. So now we're able to start trying to cure infections. And so scientists started looking at, well, this disease is caused by this bacteria. What will kill off this bacteria? Paul Ehrlich was one of the first individuals to develop a treatment for a disease. He developed Salvarsan in 1910 that was able to cure the primary stage of syphilis. In 1929, Alexander Fleming discovered penicillium mold, and what he noticed was that where the mold was growing on his petri plate, staph were being repelled. They were not able to grow in that area. And so he was able to use that, and along with two colleagues, Howard Florey and Ernest Chain, they were able to purify penicillin for use in the 1940s. And they won the Nobel Peace Prize in 1945 for this. So this drug was a lifesaver. In the 60s, people thought that penicillin would cure everything. They were giving penicillin for viral infections, bacterial infections, any infections, and it seemed to be working. But like everything, it didn't keep working, because when you overuse an antibiotic, bacteria will evolve. And so they did. They were like, ooh, there's an antibiotic. We're dying. This is going to kill us. Oh, hey, my offspring are a little bit more resilient. They have special genes that can protect them. And so now we're coming up with we're finding these problems such as antibiotic resistant bacterial species. Not only do we have antibiotic resistant bacteria, and so we're getting all of these infections that used to be easily treated are now coming back in a plethora, but we have to worry about bioterrorism. So we can actually start fighting off bacterial infections, but now with in the world we live in, we have to worry about these same pathogens being used as bioterrorist weapons. Another challenge that we're facing is climate change. So as the temperatures are warming, we're struggling to keep diseases down. So certain diseases are spread by bacteria or by insects, and with higher temperatures, we have more insects. I'm stopping now because it is almost time for this to end. So I will say goodbye and have a great night.